Okay, fantastic. So thank you very much for inviting me and giving me the opportunity to give this presentation. So I will be talking about some work that I've been doing whilst I was at the University of Warwick before I came to Oxford. So it's a very biological project, and there will be quite a bit of biology before I get into some of the mathematics and statistics. So just to give you the context, so hopefully that will be interesting for you. Okay, so the project is about the biological clock, because you can sort of see on the screen that there are all sorts of different processes, such as the body temperature, the level of alertness, the amount of melatonin or cortisol, different hormones, and for example, blood pressure, that are very much time dependent and that are oscillatory during the 24-hour cycle. You could ask the question, what stands behind this rhythmicity? What is it that sets this rhythm? And so basically, if you look at the cells individually, then there are certain genes that could be called clock genes that oscillate in let's say, their presence, their abundance. I will talk about the details so you understand more in a second. And in fact, it's these genes, their expression during a 24-hour day cycle that governs these processes being rhythmic. So, okay. What basically, there is the central dogma of molecular biology that says that there is DNA replication, and which leads to translation and production of mRNA in particular. And when mRNA is produced, so it's the mRNA that carries the genetic information that's necessary for protein production, and it's the proteins that do most of the work for proper organ and tissue functioning. And the question that we have posed is, can we put a number on the clock disruption? Because if you have cells that are somehow broken in like on the genetic level in terms of the clock, then you are very prone to problems such as getting cancer and all sorts of other diseases. And it would be really good if you could just quantify the amount of disruptiveness in order to make inference such as, right, so how much of a specific drug should we give to a certain person, or maybe what time of the day is the best for this specific individual in order to administer some sort of intervention to improve their health. So of course, so the clock that's inside each of the individual cells in different tissues, say liver, lungs, heart, and all sorts of other parts of the body. So they are the clocks that are on the periphery. And there is the master clock in the brain, the suprachiasmatic nuclei, which sort of acts as a synchronizer. And that happens through light entering the eyes and that signal going into the SCN. And basically the SCN clock being linked to the light-dark cycle that we observe because of the Earth's rotation, that sort of enables to synchronize the clocks on the periphery. However, the clocks on the periphery, meaning inside different organs such as lung, etc. If you put a person or a mouse, whatever organism you're talking about, into a completely dark environment, the rhythm will persist in those clocks through the gene rhythmicity. It just might shift a little bit because there is no entrainment via the SCN. And yeah, so I found it's a very exciting project to work on. So thinking about circadian rhythm dysfunction and disruption. So what can cause disruption to the cellular clock? So the pathways include stress, sleep deprivation, it could be pain, shift work is very prone to problems with the clock, which leads to many different problems with health, and there are some others as well. And so one of the possible results of the circadian rhythm disruption is basically gene mutation, which can lead to dysfunctionality. Yes, and I have briefly mentioned that one of the possible treatments is chronotherapy. And chronotherapy, apart from thinking carefully about what time a specific medicine should be administered in order to target the rhythm of, for example, the clock genes or the proteins that are closely connected to the genes that. Apart from that, the chronotherapy could be timed exposure to external entrainers. So basically, the idea would be to sort of try to bring the rhythmicity of the genes back, with the hope that this is going to help with the coping strategies of the organism. So yes, circadian clock is tightly coupled to different cancer hallmarks, and cancerous cells display particularly aberrant core clock gene expression. Yeah, and we would like to quantify clock disruption, and that's exactly what time teller does. And of course, it would be very good if we could put a number on the clock dysfunctionality by looking at a single biopsy, not by looking at the time series of biopsies, because it's not quite reasonable to even take, I don't know, even 3 biopsies for a person evenly spread out throughout the day, because that's quite stressful for the body, for the mind and everything. So we want to put a number based on a single timestamp. So basically, when the biopsy is taken, we would like to take the genetic information out of there. So for every single gene, we would have a read, we would have a number, and from that vector, we would like to say, right, so this person's clock disruption can be quantified as some number between 0 and 1. So time teller takes some, let's say, healthy training data from healthy individuals to build sort of like a healthy clock model. So once that model is built, we'll talk about this in detail in the next few slides. We estimate the sampling time using a probabilistic approach for a new test sample for the individual we are interested in. And we assess the uncertainty of this time estimate. And that's actually one of the problems with such data, that when doctors obtain biopsies, they very often do not record the time that the biopsy was taken. And this is quite a huge disadvantage for bioinformaticians who are interested in sort of, doing something with the information. But TimeTeller sort of puts time on the biopsy as the first step. So yes, you quantify the uncertainty of the time estimate, and the imprecision score is basically the clock dysfunctionality metric. So the hypothesis that TimeTeller is built on is that if we have very high precision of the time estimates, then it's a good clock. And if we have low precision, then it's the clock is functioning badly. So you'll see some pictures way later that will make this clear. So we've dealt with several different kinds of data sets, and also there are different platforms, there are different ways to obtain this genetic data. There's the microarray technology, there is RNA-seq technology, there is single cell technology, which we haven't worked on in this project, but we focused on microarray and RNA-seq data, and we've shown that it's possible to use it on both. So for example, the first data set we looked at was this Zhang et al. The training data sets on mice. So the mice were entrained to a specific light dark cycle, being fed ad libitum freely. And we got data for every 2 hours for microarray, and for RNA-seq, the data that we have is every 6 hours around the clock for 2 days. And so the paper itself, the authors in the paper, they focus on gene rhythmicity analysis across different tissues. And I do want to mention that 43% of protein coding genes were detected to have a circadian rhythm in at least one organ of the body. And yes, I do also want to mention that quite a lot of the drugs that are out there in the pharma that are just sold freely to people, lots of them, a huge percentage actually targets clock genes. Okay, so what do we do with this data, this training data set? So we have 8 different organs, and we look at the core clock genes. In fact, when I say core, that means that I'm taking the genes that are rhythmic in, that are commonly rhythmic in the majority of tissues, because if you take a specific organ, for example, lung or something else, there could be a different gene that is very specific to lung that also contributes to the clock, the biological clock of lung. But we only take the core ones. And so here we've got 12 different genes, and a color corresponds to a tissue. However, if you look at this data sort of, the way it is at every single gene individually, it's kind of, well, it's much harder to analyze it like this in the high dimensional feature space. So what we do is we reduce the dimensionality of the data. And that means that basically for every single timestamp that we have in a training data set, we build its own local principal component space. And therefore, if we have 4 different timestamps, then we reduce dimensionality 4 different times, 4 times, and we get 4 different spaces in which we visualize data. So every single time point, every single point that you see here, it's a single biopsy visualized in this space. So you've got multiple, because you've got multiple tissues. And so once you have that, once you've projected your data into local principal component spaces, you build the model. You do that for every single local PC space. You build the model, which basically means you find the mean, you find the standard deviation, the variance, and you also say, why don't we model this as a Gaussian distribution? And you sort of do interpolation of the means and the variances. This way you obtain sort of the model. And when you have test data, you finally discretize your time, you finally discretize your model, and you look at specific. So every single point here, like the cross section here, it would be a specific timestamp. So you take a fine discretization of your circular timeline around the 24-hour loop, and you assess the likelihood of a test point belonging to the specific timestamp, as in the specific Gaussian distribution. And you do this for every single local PC space. You obtain your likelihoods, and then you average those likelihood curves, and then you consider that average likelihood versus a specific threshold, a specific log dysfunctionality threshold that I will talk about, and then basically the measure of theta, which is what we call the dysfunctionality metric. It's very closely related to the confidence interval for the predicted time, the maximum likelihood time. High theta would be very dysfunctional clock, and that would be, we have a very large confidence interval. So here I have this all written down. So we use SVD to obtain time-specific principal components, singular value decomposition, fit Gaussian distributions in these spaces, fit a smooth spline through the times for which we have data in the training data set, extend the means and all elements of the covariance matrices, get continuous mean and the covariance matrix, take the new test biopsy data, calculate the likelihoods, average them, and select the time that gives the maximum likelihood as the time that the biopsy was taken. And then after that, what you do is, so basically, for example, here, imagine this is your average likelihood curve. And basically, well, okay, let's say normalized, because you divide by the maximum. And basically you look at the area between this and this threshold function. So in a standard statistical test, you would probably take some constant and assess whether your test statistic is greater or equal to a specific number. But in this case, instead of having a number, we have a function, which is a cosine function. And yeah, so let me talk about why we chose a specific way to look at this. So why does it help to have like a cosine function here, and that has its phase? When biologists talk about phase in relation to gene analysis, they mean the time when the maximum happens. So you sort of align the threshold function with the maximum of your likelihood curve. And imagine you had two peaks, because it's possible to have two different peaks. Imagine you have a peak here and you have a peak 12 hours out. you have another very high peak. So that would mean that you are actually very uncertain what the real time of the sampling of the biopsy is, because you would have the area above this threshold. It wouldn't be very small. It would be this, but the higher this peak, the higher the total area above the threshold. And therefore, having a 12-hour out peak here, a very big one, it would mean we have a very, very high theta. And similarly, if you have a very small peak, if you have another peak here, even if it's very high, it's not really going to make a huge difference. So yeah, if you have a wide peak, then you would also have a very low confidence, because having a wide peak would mean that, so basically the wider the peak, the higher the theta. And for us, high theta is high dysfunctionality. If you have a sort of a uniform likelihood function with one or low peaks, then you would also have low confidence. So for example, this would be an example of a very strong clock. You would be quite confident that this data point belongs to the blue distribution. And here you would be very uncertain, and you are very more likely to have, you probably will have quite a wide, I think this looks like a uniform likelihood function on the average. So yes, so that's the gist. And we took several different data sets. We didn't just look at mouse data, we looked at human data and baboon data, and we basically analyzed all sorts of data sets and built software that allows to very quickly do analysis. So this specific paper from which we took as our test, it focused on the lack of food, effect of the lack of food on the functionality of the clock, and it was on mice. So we had data for every 4 hours. Here it says starting at ZT0, that's sort of a biological term for Zeitgeber time. If you have mice that are sort of have a specific condition for the light and dark, then ZT0 is the time when the lights go on. And if instead of ZT you see CT in my slides, that means it's the circadian time, which means that the animals, or whichever organism is under consideration, is under free running conditions, meaning that they don't have a specific condition for analysis. So, I mean, the paper sort of focused on, they mostly analyze genes individually. And one of the things that they say is that fasting does enforce specific temporal gene regulation, and like there is this gene ANTL and REVAB alpha, which are very important for good clock functionality, and this is exactly what the authors observed, that fasting enforces blunted rhythmicity of these genes in liver and skeletal muscle. However, what we did was we looked at this data in our context, let's say. And if you look at the mice that were fed ad libitum, meaning they weren't starving, they were fed freely, then you can see that if we built the model using that Zhang data set that I talked about in the beginning, this is RNA-seq data. If you build that model and you put the this Kinushi data, ad libitum data on it, you can see that it falls very nicely on the model. Whilst if you take the data for the starved mice, you can see that not only does it fall outside of the model, it also, you can sort of even see sort of a different ellipses, let's say. So that is very interesting, because that sort of means that the clock is still sort of functional, but it's just off. And yeah, it would be interesting to put a number on this functionality. So that's exactly what we did. So we considered the difference between the fed and starved mice groups. And for the ad libitum mice, the thetas were very low, whilst for this is skeletal muscle, by the way, the tissue that's under analysis. Whilst if you look at starved mice, also higher thetas, and there is a significant difference between the two groups. And also, if you look on the plots on the left, so here the colors of the points. So if you see blue being close to the blue line, that's good. That means that the time predicted was blue was close to the, to where it should be. However, on the y-axis you have the theta. So and you can see here that the squares are the mice that are fed ad libitum, and they are generally lower than the totals, which correspond to the mice that are starved. So that's one of the examples. Another example that we looked at, so here we have mice which are, so we've got wild type mice, which means sort of freely fed, nothing was done to them, no specific gene modifications or everything like that. So, and you have mutant mice which have a knockout of a specific gene, reverb alpha, that I mentioned earlier. That's very important for good functionality of the clock generally. And knockout is basically means you sort of, it doesn't function. So yeah, if we look at this paper, one of the major points again is that reverb alpha gene is important for proper transcriptional control of clock function. So when I say transcriptional, that's related to mRNA expression. Yeah. So if we look at that data set, and if we put the test data sets points onto the model, here is actually, this is the microarray data, the Zhang microarray data that's used as training. We see that there is a difference between the two groups. So the reverb alpha knockout mice, they sort of fall more outside of the model, whilst the wild type mice are very nicely just bang on the model. And if we look at the thetas, we see there is quite a large difference between the theta groups, a significant difference. And also, if you look at the predicted time, or what can you say? You can say that for the wild type, they're sort of more bunched around the time when the biopsies were taken. Well, yeah, when the data was taken, let's say. So that's quite good. I mean, these data sets, they are completely independent of the training data sets, and the data was generated by completely different labs, and therefore it was interesting to look at how the data sets combine and what we're able to learn having like a model from healthy individuals, healthy organisms.
I'm really sorry, but you have 1 minute left.
Okay, fantastic. So we also looked at data that's related to sleep deprivation. And we were able to sort of look at that and make some inference from this. We looked at human data, and we sort of get nice models, and we've used that to analyze cancer patients. And theta can be used as a potential prognostic biomarker in cancer studies, completely independent of the general standard biomarkers that are out there. So to conclude, time teller may be used to microRNA and RNA-seq technology, and actually you can combine the two in exactly the same model without much modification. You can use this to detect changes to circadian clock functionality from single biopsies. Time teller is able to pick up type differences between individuals, and potentially this is useful for current, mostly people from the University of Warwick and in France. So thank you very much for your attention, and I'm happy to answer your questions.
No.